Imagine London in the 1920s, a bustling city full of life and energy. The Great War was over, but its shadows lingered. People were looking for new beginnings, new hopes. Science too was on the move. Discoveries were changing the way we understood the world. In the heart of this vibrant city stood St. Mary's Hospital, a place of healing and also a place of research. Doctors and scientists worked tirelessly. They were searching for ways to fight diseases that, in those days, could often mean a death sentence. Inside St. Mary's, there was a laboratory. It wasn't the grandest or the most modern, but it was a place where important work was happening. This was the domain of Dr. Alexander Fleming, a bacteriologist, a scientist who studied tiny living things called bacteria. Some bacteria are harmless, even helpful, but others, as Fleming knew all too well, were dangerous killers. They caused infections that doctors struggled to treat. Fleming was a quiet, observant man. He had served in the Royal Army Medical Corps during the First World War. He had seen firsthand the terrible impact of infected wounds on soldiers. Fleming's world was one of petri dishes and microscopes. He spent his days growing bacteria, studying their behavior. He was looking for something, anything that could stop them. The quest for a weapon against these invisible enemies was a constant driving force in laboratories like Fleming's. Alexander Fleming was a brilliant scientist, but he was not known for his tidiness. His laboratory was often quite cluttered. Stacks of petri dishes containing various cultures of bacteria would often be left on his workbench. Fleming was not one for meticulously cleaning up every experiment immediately. This habit would prove to be incredibly important. In the late summer of 1928, Fleming was preparing to go on holiday. Before he left, he had been working with Staphylococcus. He had a number of petri dishes where these bacteria were growing. As was his custom, he didn't meticulously clean and sterilize all of them before departing. Some were stacked in a corner of the lab, perhaps near an open window. Fleming went on his holiday, leaving behind his experiments. A tiny airborne spore of a particular type of mold, a Penicillin mold, had drifted in and settled on one of the dishes. Upon his return from holiday in September 1928, Fleming began the task of sorting through the clutter in his lab. He started to examine the old petri dishes he had left stacked up. Many were simply overgrown with bacteria, as expected. But then, his sharp, observant eyes noticed something peculiar on one particular dish. It was a dish that had been contaminated with a blue-green mold. Around the patch of mold, the colonies of Staphylococcus bacteria looked different. They appeared to have been killed. There was a clear ring, a sort of halo, around the mold where the normally opaque bacterial colonies had vanished. Further away from the mold, the bacteria were growing normally. It was as if the mold was producing something that was stopping the bacteria from growing or even destroying them. This was the moment of true discovery. Fleming realized this was more than just a simple contamination. This mold, Penicillium notatum, seemed to possess a remarkable power. He took a sample of the mold and began to cultivate it. He wanted to understand what was happening. Fleming began to experiment with his newfound mold. He grew it in liquid broth. He found that this mold juice, as he sometimes called it, was incredibly effective at stopping the growth of many types of harmful bacteria. Not just Staphylococcus, but also Streptococcus, Pneumococcus, and the diphtheria bacillus. The potential was enormous. Here was something produced by a simple mold that could kill some of the most feared bacteria of the time. He also discovered something else very important. The mold juice, while deadly to bacteria, appeared to be harmless to animal cells. This was a critical finding. Many antiseptics known at the time were effective at killing bacteria, but they also harmed human tissue. This new substance from the Penicillium mold seemed to be different. It was selective. It targeted the bacteria, leaving animal cells unharmed. Fleming named the active substance penicillin, after the Penicillium mold that produced it. He published his findings in the British Journal of Experimental Pathology in 1929. Section 5. The Oxford Torchbearers, Florey and Chain take up the quest. Nearly 10 years passed. Fleming's discovery of penicillin, while remarkable, had not yet led to a usable drug. The challenges of isolating and purifying the active compound were significant. But the memory of that bacteria-killing mold lingered in the scientific literature. Then, in the late 1930s, a new team of researchers at the University of Oxford turned their attention to it. This team was led by Professor Howard Florey, an Australian pathologist, and Dr. Ernst Chain, a brilliant Jewish biochemist who had fled Nazi Germany. They were joined by Dr. Norman Heatley, a biochemist with a remarkable talent for improvisation and ingenuity. Florey and Chain were systematically investigating naturally occurring antibacterial substances. They came across Fleming's 1929 paper on penicillin. They were intrigued by its potent effect and its apparent non-toxicity to animal cells. The world was also changing. The threat of another major war was looming in Europe. The need for effective treatments for infected wounds, a major killer in wartime, was becoming increasingly urgent. This provided a powerful motivation for their research. They decided that penicillin was worth a closer look, despite the difficulties Fleming had encountered. They believed that with advances in biochemistry, they might succeed where Fleming had not. Section 6. From Laboratory Bench to Life-Saving Trials. The year was 1940. The Oxford team led by Florey and Chain had finally produced enough of their precious brown penicillin powder to test its effects on living creatures. The first critical experiments involved mice. They infected 8 mice with lethal doses of Streptococci bacteria. Then, they treated 4 of these mice with penicillin. The other 4 were left untreated as a control group. The results were dramatic, almost miraculous. Within hours, the untreated mice were gravely ill, and by the next morning they were all dead. But the 4 mice that had received penicillin were alive and well. It was a stunning success. This experiment provided the first clear proof that penicillin could work inside a living body to cure a deadly bacterial infection. It was a pivotal moment. The team was elated, but they knew there was still a long way to go. Producing enough penicillin even for these mouse experiments had been a huge effort. To treat a human would require hundreds of times more. They scaled up their makeshift production line. They used every available container, including hospital bedpans and biscuit tins, to grow the penicillium mold. The entire Dunn School of Pathology at Oxford was transformed into a small penicillin factory. Section 7. The Crucible of War Penicillin Goes to Battle. In the summer of 1941, Howard Florey and Norman Heatley traveled to the United States. Their mission was urgent: to persuade American pharmaceutical companies and the U.S. government to help mass-produce penicillin. Britain was under siege, its resources stretched to the limit by the war. They carried with them not a large supply of penicillin, which was still incredibly scarce, but something far more valuable: the penicillium mold itself and the knowledge of how to extract its precious product. They arrived at a critical time. The U.S. was not yet officially in the war, but the government was beginning to prepare for potential involvement and understood the medical needs that would arise. Their initial meetings were met with some skepticism. American companies were already investing in sulfa drugs. Producing penicillin on an industrial scale presented enormous technical challenges. The mold produced very little of the active substance. It needed to be grown in specific conditions. The extraction process was complex. However, scientists at the U.S. Department of Agriculture's Northern Regional Research Laboratory, NRRL, in Peoria, Illinois, took up the challenge with enthusiasm. Section 8. A World Transformed, the Antibiotic Dawn, and a Prophet's Warning. The end of World War II saw penicillin become widely available to the civilian population. Its impact was nothing short of revolutionary. Diseases that had once been terrifying killers, pneumonia, scarlet fever, syphilis, gonorrhea, diphtheria, rheumatic fever, could now be treated effectively. Surgical infections, a major risk in any operation, became far less common. Childbirth became safer. The average human lifespan began to increase significantly, partly thanks to this remarkable mold. Penicillin ushered in the antibiotic era. It was the first true antibiotic, a substance produced by one microorganism that could kill another. Its success spurred a gold rush to find other antibiotics. Scientists began to screen thousands of soil samples from all over the world, searching for other microorganisms that produced antibacterial substances. This led to the discovery of streptomycin in 1943, effective against tuberculosis, followed by chloramphenicol, tetracyclines, and many others. A whole new arsenal of weapons against bacterial diseases became available to doctors. The world was transformed. The fear that once accompanied common infections began to fade. Section 9. The Enduring Echo, Curiosity, Chance, and the Future of Healing. The story of penicillin is a wonderful testament to the power of scientific curiosity. Alexander Fleming wasn't looking for an antibiotic when he returned from his holiday. He was simply an observant scientist who noticed something unusual, something funny, and decided to investigate it further. His mind was prepared to see the significance in an accidental contamination. This highlights a crucial aspect of scientific discovery, the importance of being open to the unexpected. Sometimes the greatest breakthroughs come not from a meticulously planned experiment, but from a moment of serendipity, a fortunate accident, combined with a keen eye and an inquisitive spirit. The journey of penicillin from a moldy petri dish to a mass-produced drug also shows the vital role of collaboration in science. Fleming made the initial discovery, but it took the dedicated work of Florey, Chain, Heatley and their team at Oxford to isolate and test it. And then it required the industrial might and ingenuity of scientists and companies in the United States to make it available to the world. Each step was essential. Science rarely progresses through the efforts of a lone genius. It is often a cumulative process with different people with different skills building upon each other's work. This collaborative spirit is more important than ever today.